I, Pooja Verma, welcome you all to this session. Hope all of you are fine and doing great. In today's session, we will learn about biomacromolecules, various inorganic constituents present in the living tissue, monomeric units of proteins, carbohydrates and nucleic acid. We will discuss the structure and function of lipids. Learning objectives of today's session are to help you learn about various biomacromolecules found in living organisms, to help you understand the concept of fresh weight, dry weight and ash, to compare the percentage of various elements present in non-living and living matter, to help to learn about the various inorganic constituents of living tissues, to learn about the monomeric units of proteins, carbohydrates and nucleic acid, to help you understand the structure and function of lipids, to develop scientific attitude, creativity, aesthetic sense and problem solving approach in you, to enable you to find applications of these topics in your daily life. We know that the number of species that are known and described range between 1.7 to 1.8 million. Such a huge biodiversity exists on our planet Earth. Now a question that arises in our mind is, are all living organisms made up of same chemicals? To find the answer, we have to take a living tissue. We can take either a vegetable or a piece of liver and grind it in trichloroacetic acid. I hope all of you know the formula of acetic acid, which is CH3COOH. If we replace the 3 hydrogen atoms in acetic acid with 3 chlorine atoms, we will get trichloroacetic acid. By using this trichloroacetic acid, grind the tissue using a pestle and mortar to obtain a thick slurry, filter it using a cheesecloth or cotton, we will obtain 2 fractions. The first one is the acid soluble pool, which contains biomolecules and their size is less than 1000 Dalton. These micromolecules may be inorganic or organic. The other fraction is acid insoluble pool, which contains biomacromolecules, which are large sized complex chemicals having high molecular weight, usually more than 10,000 Daltons, except lipids. They are always organic macromolecules. They generally belong to 4 classes of organic compounds such as carbohydrates, proteins, lipids and nucleic acids. Inorganic elements and compounds are also present in living organisms. How can we find them? For doing this, first we need to weigh a small amount of a living tissue. We can use either a leaf or a liver and take its weight. The weight will be called wet weight. Now we have to dry the tissue so that all the water evaporates. Take its weight again. This weight will be called dry weight. Now we need to burn the tissue completely so that all the carbon compounds are oxidized to gaseous forms, that is carbon dioxide and water. The remaining part is called ash. This ash contains inorganic elements. These inorganic compounds are seen in the acid soluble fraction. If we compare the elements present in non-living and living matter, we will find that human body contains large amount of carbon, hydrogen, oxygen and nitrogen compared to Earth's crust, as you can also see in the table. Now let's see what all inorganic constituents are present in living organisms. As you can see in the table, living tissues contain inorganic constituents like sodium, potassium, magnesium, water and compounds like sodium chloride, calcium carbonate, phosphate ions, sulfate ions, etc. We will now discuss in detail about biomacromolecules. Biomacromolecules such as proteins, lipids, nucleic acid and carbohydrates are formed by the polymerization of monomers, which are the simplest units. As they are formed from monomers, by polymerization they are called polymers. For proteins, amino acids are the monomeric units. Protein is a macronutrient that is essential for building muscle mass. It is commonly found in pulses, fish, eggs, nuts and seeds. Proteins are formed by joining of amino acids through peptide bonds. You can see the general structure of amino acid on the screen. Amino acids are substituted methane in which 4 hydrogen atoms are replaced by carboxyl group COOH, amino group NH2, hydrogen and a variable group which we call R group. Amino acids are called alpha amino acids as the central carbon is alpha carbon, which means a carbon in which 4 substituents group occupy the 4 valency positions. Now let's discuss about the R group. It could be simply hydrogen as in case of glycine, which is the simplest amino acid. R could be a methyl group as in case of alanine. It could be a hydroxyl methyl as in case of serine. 20 types of amino acids are found in proteins. Now based upon the number of amino and carboxyl groups, we can classify amino acids into 3 categories. The first one is the neutral amino acids. They have 1 amino group and 1 carboxyl group. Amino group has +1 charge, carboxyl group has -1 charge. So they neutralize each other and the overall charge on the amino acid is 0. Such amino acids are called neutral amino acids. Example is glycine. Second category is acidic amino acids. If the R group has carboxyl group, then this amino acid will have -2 charge, one from the carboxyl group of R chain and one from the carboxyl group, and +1 charge due to amino group. Thus, the overall charge on the amino acid will be negative. Such amino acids are called acidic amino acids. Example is glutamic acid, in which value of R is CH2COOH. Try to draw the structure in your notebooks. The third category which we have is basic amino acids. In this case, the R group contains an additional amino group. Now try to calculate the number of positive charge present in such an amino acid. Let's take the example of lysine, in which R is CH2, 4NH2. Such amino acid will have +2 charge, one from the amino group, one from the R group and -1 charge from carboxyl group. Overall charge on the molecule will be +1. Such amino acids which have overall positive charge on them are called basic amino acids. As we have just discussed, the example is lysine. Some amino acids are aromatic amino acids such as tyrosine, phenylalanine, tryptophan. The R group in them contains an aromatic ring. Now let's discuss about the ionizing property of amino group and carboxyl group. Both these groups are ionizable in nature. Hence, in solutions of different pH, the structure of amino acid changes. As you can see on the screen in figure A, amino group is present in ionized state. The overall charge on the amino acid is positive. In figure C, carboxyl group is present in ionized state and the overall charge on the amino acid is negative. Now see diagram B carefully. In this case, both amino group and carboxyl group are present in ionized state, +1 charge due to amino group and -1 charge due to carboxyl group. This structure is called Zwitter ionic structure. Zwitter ions possess both positive and negative electrical charges, but their net charge is 0. Every amino acid acquires Zwitter ion structure at a particular pH called isoelectric pH. Now let's discuss about lipids, which are generally water insoluble. You have noticed that if you try to mix oil with water, oil never dissolves in water. So lipids are fatty acid esters of alcohols. They are made up of fatty acid and glycerol joined with ester bond. You have studied about esterification reaction, in which acid and alcohol join with each other with the help of ester bond. Now let's look at the structure of glycerol, which is trihydroxypropane. As you can see, This compound contains 3 carbons and 3 hydroxyl groups. It is a simple lipid. Lipids could also be simple fatty acids. Fatty acid has a carboxyl group attached to an R group. General structure of carboxyl acid is RCOOH. R group could be hydrogen, methyl or ethyl in case of methanoic acid, ethanoic acid and propionic acid. Plants contain organic acids in which value of R is 16 to 20 carbons. These are called fatty acids. Count the number of carbons present in palmitic acid. As you can see, it is 16. Depending upon whether fatty acids contain single bonds or double and triple bonds, we can classify them into saturated and unsaturated fatty acids. As you can see the structure of palmitic acid on screen, it contains only single bonds, thus it is a saturated fatty acid. Focus on the structure of oleic acid. It contains 18 carbons and double bonds, thus it is an unsaturated fatty acid. Many lipids have both glycerol and fatty acids joined with the help of ester bond. If one fatty acid is attached with glycerol, through ester bond it forms a monoglyceride. Mono means one. If two fatty acids are attached with glycerol through ester bond, then it is called a diglyceride. Di means two. The word triglyceride refers to 3 molecules of fatty acids esterified to a single molecule of glycerol. 3 molecules of water are eliminated in the process. You can see the ester bond inside the red circle. The ester bond is O, C double bond O. On the basis of melting point, lipids are also called fats and oils. Fats have higher melting points and they are present in solid state at room temperature. Example is butter, which is consumed regularly by many people. Another example is dalda ghee. On the other hand, oils have lower melting point. They are present in liquid state at room temperature even in winters. Examples are groundnut oil, mustard oil. You might have noticed in your homes that even during severe winter, mustard oil remains in its liquid state. The low melting property is actually the property of fatty acids. Oils are richer in unsaturated fatty acids. Oils with polyunsaturated fatty acids, that is fatty acids with more than one double bond, are called polyunsaturated fatty acids, PUFA. They are commonly recommended by doctors or physicians to persons having high blood cholesterol, heart diseases, because polyunsaturated fatty acids lower blood cholesterol. Some lipids contain phosphorus and phosphorylated organic compounds in them. These are called phospholipids. They are triglyceride lipids. One fatty acid is replaced by phosphoric acid, which is often linked to additional nitrogenous group. They are present in the plasma membrane. Recall the structure of plasma membrane. It is made up of lipid bilayer and proteins. This lipid bilayer contains phospholipids. Lecithin is an example of phospholipids. Phospholipids are amphipathic. Head is hydrophilic, that is, likes water, and tail is hydrophobic, that is, it repels water molecules. Lipid bilayer is the basic component of all plasma membranes. Cholesterol is a common sterol found in many animals including humans. It is a lipid with unique structure. It consists of 4 hydrocarbon rings forming the bulky steroid structure. There is a hydrocarbon tail linked to one end and a hydroxyl group linked to the other end. Cholesterol is called sterol because it is made up of alcohol and steroid. Cholesterol can be considered as our friend as well as foe. Cholesterol is the raw material needed to form steroid hormones like progesterone, estrogen, testosterone, aldosterone, cortisol, etc. Thus, it is useful to us. But when cholesterol level rises in the blood due to its excessive intake in our diet, it gets deposited in the wall of arteries, thereby blocking them. This causes high blood pressure and many heart diseases. Now let's discuss about carbohydrates or polysaccharides. They are commonly called sugars. They are sweet in taste, soluble in water. Their general formula is CH2ON. Polysaccharides are polymers. They are formed by the joining of monomeric units, that is monosaccharides with the help of glycosidic bond. Monosaccharides are those simple units which cannot be broken down further into smaller components. Glucose is a monosaccharide containing 6 carbon atoms. Its molecular formula is C6H12O6. You can see the structure of glucose on the screen. The 6-membered cyclical structure of glucose is called pyranose structure. The ring contains 1 oxygen and 5 carbon atoms. 6th carbon atom is present outside the ring in the form of CH2OH. Sugar, which we eat in our daily life, is sucrose, which is formed by the joining of glucose and fructose with the help of glycosidic bond. Ribose, a pentose sugar, is a monosaccharide containing 5 carbon atoms. The molecular formula is C5H10O5. It is found in RNA, a type of nucleic acid. The 4th biomacromolecule is nucleic acid, which can be either DNA or RNA. They are polynucleotides made by joining of monomers called nucleotides. Nucleotides are basic units of nucleic acid. Nucleotide is made up of nitrogenous base, sugar and phosphoric acid. Nitrogenous base which is present in nucleic acid could be either purine or pyrimidine. Purines are large, 9-membered double rings, as you can see the structure on screen. It has nitrogen at 1st, 3rd, 7th and 9th position. It is of 2 types, adenine and guanine. Pyrimidines are smaller, 6-membered rings. A pyrimidine ring has nitrogen at 1st and 3rd position, as you can see on the screen. It is of 3 types, cytosine, uracil and thymine, commonly abbreviated as CUT. Nucleic acids contain either ribose or deoxyribose sugar. Ribose sugar is present in RNA. Its molecular formula is C5H10O5. Deoxyribose sugar is present in DNA. Its molecular formula is C5H10O4. Both ribose and deoxyribose sugars occur in furanose state or pentagon ring with 1 oxygen and 4 carbon atoms. The 5th carbon having CH2OH complement lies outside the ring. Ribose differs from deoxyribose in having a hydroxyl group instead of hydrogen at 2nd carbon. A combination of a nitrogenous base with a pentose sugar is known as nucleoside. Observe the structure of adenosine shown on the screen. You can see that adenine nitrogenous base is linked with ribose sugar through glycosidic bond. If purine is a nitrogenous base involved in the formation of nucleoside, then the suffix sign is used, as in case of adenosine and guanosine. If pyrimidine nitrogenous base is involved in the formation of nucleoside, then the suffix -dine is used, as in case of cytidine, thymidine and uretine. So we can say nucleoside equals to nitrogenous base plus sugar. When nucleoside joins with phosphoric acid, they form nucleotides. Nucleotides are phosphoric acid esters of nucleosides. Phosphate combines with sugar molecule at its 5 carbon atom. Suffix used for nucleotide is ilic acid. So we have adenylic acid, guanylylic acid, cetidylic acid, thymidyllic acid and uridylic acid. Now these monomeric units will join together with the help of phosphodiester bonds to form nucleic acids. Let's try to understand the differences between nucleotide and nucleoside. Nucleotide is formed by the joining of nucleoside and phosphoric acid, whereas nucleoside is formed by joining of nitrogenous base and sugar with the help of glycosidic bond. Nucleotide helps in the formation of nucleic acid. It is the monomeric unit of nucleic acid, whereas nucleoside is the precursor of nucleotides. In case of nucleotides, we use the suffix ilic acid like adenylic acid, guanidinic acid, citidinic acid, thymidinic acid and uridylic acid, whereas in case of nucleoside, suffix sign is used if the nitrogenous base is of purine type. Examples are adenosine and guanosine. If the nitrogen bases are of pyrimidine type, then we use the suffix -dine. Example cytidine, thymidine, uridine. One common mistake committed by the students is that they think that cytosine is a nucleoside. Actually, it is a nitrogenous base. If cytosine forms nucleoside, since it is a pyrimidine base, we have to use the suffix -dine. Thus, its nucleoside will be called cytidine, not cytosine. Here is a quick recap of what we have learned today. We have learned in detail about the various biomacromolecules found in living organisms. We have also understood the concept of fresh weight, dry weight and ash. We have learned about the various inorganic constituents of living tissues and also the monomeric units of proteins, carbohydrates and nucleic acid. We have also understood the structure and function of lipids. Before we conclude, here is an assignment for you. You might have heard about PUFA in various TV ads of refined oil. Write the full form. Why are they considered good for heart? What is the benefit of eating proteins? How much proteins should an adult consume in one day? Can excessive consumption of protein health supplements be harmful, especially for kidney patients? Why excessive consumption of sugars should be avoided? Collect information on why it is said India is the diabetes capital of the world. Suggest ways how we can keep ourselves healthy and fit. Tabulate 4 major differences between purine and pyrimidine. I hope you all enjoyed the session. Keep smiling, keep learning. Thank you.